Good afternoon, everyone. Hopefully you can all hear me. I'm Liz Otteson. I'm an associate professor in microbiology at UGA. My background is um, in microbiology and microbial community ecology in particular, but over the years I've become increasingly interested in the ecology of antibiotic resistance, which is how I ended up as part of this group. So my goals for today is just to provide a high-level overview of the problem and particularly terminology, concepts, and concepts, and approaches that are commonly used in understanding the molecular epidemiology of antibiotic resistance to hopefully help you get perspective on the talks and to help you think about what tools you might want to implement in your own research or what tools you might need to understand other people's work. So, given the audience, I don't feel like I need to spend a ton of time convincing you that antibiotic resistance is an important problem. So, according to the 2019 report, we were at about close to 3 million infections with antibiotic-resistant bacteria or fungi per year in the US, which are responsible for somewhere around 36,000 deaths each year. That was in 2019. We then had the COVID-19 epidemic, which resulted in increases in a lot of different antibiotic-resistant organisms, although the most recent report suggests that things might be starting to level off again. So it's a major problem. And as part of the perspective, many of you may have seen this graph as well, which is a sort of timeline of major antibiotic releases on the left, starting with penicillin in 1943 and ending with ceftaroline in 2010. And the time between that and when there were the first literature reports of antibiotic resistance to that antibiotic. And, this is one of those timelines that's shown a lot, mainly for 2 points, one of which is that the rate of new releases has not been as fast as we would like to see. And the speed at which we start seeing resistance is frighteningly high, and in some cases, and possibly even increasing. So, what lies at the root of this problem? One of the major things is that when designing antibiotics, they're a poison, right? And your big goal is that you find something that'll poison bacteria without poisoning us. And as a result, we're kind of limited in targets to things that are essential to bacterial cells, but sufficiently different in bacteria than they are in humans that we can target them in a lot of bacteria, make it hard for the bacteria to rewire around whatever process we're targeting, while at the same time having it sufficiently different from the eukaryotic systems, our own systems, that we aren't poisoning ourselves. Which is also why I'm not going to spend a ton of time on antifungals, but it's the major reason why there's a lot fewer antifungals out there than there are antibiotics. So we're kind of limited in our targets. Major antibiotics tend to target cell wall synthesis, certain processes within DNA and RNA synthesis, folate synthesis, cell membrane production, and protein synthesis. And the problem is we have a limited number of targets, and bacteria have a lot of different ways of getting around our efforts. So some of the major forms of antibiotic resistance are things like efflux, where they just pump your antibiotic back out of the cell. You have immunity and bypass, where you rewire whatever process you're targeting so that your antibiotic no longer targets it effectively. Or you come up with a new pathway that essentially goes around the step that you're trying to block. Or just straight up immunity where whatever a particular bacterium is doing is sufficiently different from the target for the antibiotics that it just doesn't hit. You have target modification, where you modify whatever process thing your antibiotic is targeting to keep it from hitting. And then inactivating enzymes, which might chew up the antibiotic and render it non-functional. And so you have a limited number of targets and a lot of ways to get around them. And on top of that, most antibiotics actually have their roots in microbial warfare. They are based on natural products that have been adapted or modified for use in medical situations, which means that oftentimes something similar, if you have a similar antibiotic already out in the environment, you also have, in most cases, resistance already out there, where microbial warfare is permanently ongoing. It's a fundamental part of microbial community ecology. So when you steal things from that to try and use for medical purposes, you have to take into account the fact that somebody out there probably has already figured out how to get around that. And then on top of that, every time you use antibiotics, you're imposing selective pressure that then favors the growth of antibiotic-resistant organisms. So there is essentially no way of using antibiotics in a way that prevents the generation of antibiotic resistance. There's various things that we do to minimize that, using your full course of antibiotics, only using your most valued antibiotics when you have no other choice. But to some extent, every time you use an antibiotic, you're already devaluing it to some extent. And then bacteria make our lives even harder through horizontal gene transfer, which is that resistance doesn't just stay in one organism. So one of the points I like to make early on is that horizontal gene transfer is not a fluke. It is not an accident. It is not a sometimes thing. The reality is that horizontal gene transfer is a fundamental aspect of microbial biology. Virtually every group of bacteria, to some extent, will undergo horizontal gene transfer. And if a bacterial population becomes sufficiently isolated that it is unable to undergo horizontal gene transfer, for example, in many endosymbionts, in the long run, it tends to result in an evolutionary dead end. So it's just like essentially every higher animal has sex. There are very few animals that reproduce without sex because you need to be able to change your DNA to adapt to the environment. There's Mueller's ratchet, the idea that deleterious mutations, if they don't immediately kill you, can accumulate and propagate through and become fixed within the population through genetic drift, as well as Red Queen hypothesis, which is essentially that organisms are essentially permanently in a state of warfare or competition, and if your opponents can exchange genes and change up their population and can evolve faster than you can, you tend to be get into trouble. So just like for eukaryotes, essentially every animal population that reproduces by asexually, and they do arise periodically, it tends to become an evolutionary dead end. Same thing for bacteria. Bacteria never have sex, but they do exchange DNA. constantly through any number of different mechanisms, including some of the major ones are transformation, which is the simplest in many ways, which are not all bacteria, but some bacteria are naturally competent. They will just pick up DNA out of the environment and stick it into their genome through homologous recombination. You have the more deliberate approach of conjugation. It's particularly popular in gram negatives, where the bacteria will have conjugation genes, produce a sex pilus, use it to transfer usually plasmids, but plasmids can incorporate it into the genome and get passed along. They can pass along other plasmids by accident. Conjugation is essentially deliberate transfer of DNA from cell to cell. And then you have transduction, where as part of a viral infection, bacterial DNA might get passed along due to accidental incorporation into a phage package that then allows it to be inserted into another bacterial cell. And so all of these mechanisms are important for diversifying the bacterial genome, and all of these mechanisms can move antibiotic resistance genes from one organism to another. And to give you an idea of the extent to which this is a consideration, here I just went through the 2019 antibiotic resistance threats reports and put a star next to all the ones where, to some extent or another, antibiotic resistance is part of the acquisition or spread of this particular antibiotic resistance. And as you can see, it's a very large fraction of antibiotic resistance threats, organisms that for one reason or another can either acquire or spread antibiotic resistance genes through the population. We do have a few saving graces, one of which is that in practice, horizontal gene transfer tends to be restricted to, not restricted, but most common and more closely related organisms. The farther two organisms are on the family tree, the less likely antibiotic resistance is going to happen. Mainly because, first of all, you need to have, if you pass a horizontal gene transfer, if you pass an antibiotic resistance gene along, it needs to be expressed. So the promoter associated with that gene needs to be something that works with the target cell. You have to, whatever antibiotic resistance gene is being produced has to work in that organism and be compatible with the organism who gets it. You have to, bacteria, yes, they exchange genes naturally, but at the same time, they also have a lot of immune systems trying to avoid, that are trying to protect them from phage and from acquisition of harmful DNA, which means that horizontal gene transfer often has to be something that evades bacterial immune systems. And then on top of that, whatever vector you're transporting the DNA on, for example, plasmids need to have compatible machinery in the cell that allows that plasmid to be propagated, unless you have the bad luck to have that bacterial gene jump into the genome using transposons. So the reason I went through all of that is that anytime you're doing epidemiology of antibiotic resistance, you need to not just consider epidemiology of the bacterial host, but in some ways you have to consider epidemiology of the particular resistance gene you're looking at as almost its own independent entity. And then we make things even harder, which is that most pathogens are not obligate pathogens. And particularly many of the major AR threats are organisms that have substantial environmental, commensal, or commensal populations associated with them. So antibiotic resistance, we tend to focus a lot on healthcare associated infections, but the reality is that most of the bugs that are major AR threats here again, I marked them with stars, are organisms that have either environmental spread or commensal populations. All right, I saw in the chat someone was asking about transposons. I am not sure. I thought I had that slide. I may have moved it to a different part of the talk. But transposons are essentially jumping genes where it is a transposase is an enzyme that allows a small unit of DNA to move around from one area of the genome to another. Then that can also facilitate antibiotic resistance genes moving both within a cell and between the chromosome and a plasmid. All right, and then when we consider environmental spread and environmental populations, we need to consider a lot. That complicates our picture quite a bit. And it's a major part of antibiotic resistance because we have to consider not only selective pressures and antibiotic exposures within the patient, we also need to consider selective pressures that act on your bacterial population outside of a patient. So we have antibiotics being produced. One of the major ones is that those antibiotics don't necessarily we tend to think of medicines as going to people and then disappearing. But the reality is that not every part of every medicine is broken down in the body. And it leaves your body either in urine or stool, goes into wastewater treatment, which is really focused on eliminating pathogens. And a lot of different antibiotics and other medications are actually not broken down during wastewater treatment and can get out into the environment, or only partially broken down during wastewater treatment and get out in the environment. And we're finding, as we've gone to look, that vast majority of waterways, for example, are contaminated with all kinds of pharmaceuticals, antibiotics, and antibacterial agents of one type or another. And all of these can have at least low levels of selective pressure on bacteria to maintain antibiotic resistance genes. You have things like treatment of the actual production waste from pharmaceutical facilities can result in release of different compounds into the environment. You have food use of antibiotics, food animal use of antibiotics that can then result in selection for antibiotic bacteria present in food that might either directly contaminate our food or contaminate manure or waste, get out in the environment, and then contaminate us. So we increasingly also have to consider environmental pressures and their role in the spread of antibiotic resistance. And this does not play particularly well with the horizontal gene transfer problem, which is that I mentioned before, antibiotic resistant to most antibiotics are out there in the environment. Well, many of the major antibiotic resistance genes that we run into started their life as a resistance gene or a gene present in an environmental organism that may or may not have been directly related to resistance, but gets mobilized, transferred into pathogens through one mechanism or another. There's a lot of different routes by which this can take, and becomes a problem eventually. And then, of course, you get even more fun, which is that antibiotic resistance genes tend to accumulate to form multi-drug resistance, either multi-drug resistant plasmids, or bacteria will often acquire multiple resistance genes because for a variety of reasons, one of which is that maybe a gene provides resistance to multiple different antibiotics. For example, efflux pumps can pump out all kinds of different things. Or if you have two genes present that have synergy that can help, or you might have multiple genes on a single operon that are co-regulated by the same factor, produce resistance to multiple things, and then selection in one end for one of those will select also for your other antibiotic resistance. So that's how you start having problems. Going back, somebody in the chat was asking about organic food and whether it has less of a medication and antibiotic resistance issue. That's less my specialty. In theory, yes. In reality, I don't know if it is a large enough difference to be effective. I believe it is at least somewhat lower. Simply because, but on the other hand, we also aren't allowed to do very much antibiotics as growth promoters, at least. So a lot of the co-selection, I think, is indirect these days in food. And honestly, there's a lot of questions still how much the resistance in food impacts resistance in human pathogens. And there's a lot of big question marks. We know these things happen, but the scale is actually really difficult to quantify. So which is more important, antibiotic use in food, antibiotic use in humans? We don't actually know yet. All right. So the upshot here is that, antibiotic resistance, we need to consider both epidemiology of the resistant bacteria as well as separately whether the antibiotic resistance genes have their own sort of epidemiological history. And then in many cases, we should keep in mind that the vast majority of our data comes from healthcare associated expression of antibiotic resistant genes. And it is much harder to track, but important to keep an eye on what's going on outside of humans and outside of the healthcare environment that might be promoting the spread of these organisms. All right, so that's sort of the end of my lengthy introduction to get into the more technique-oriented aspects of this talk, where I'm going to be talking about, first, I'm going to be giving a quick primer on molecular epidemiology of bacteria and some of the terminology you need to be aware of, followed by information on antibiotic resistance gene identification, and then a very brief overview of mobile genetic elements and what you might be looking for to try to understand horizontal gene transfer and its involvement in the movement of antibiotic resistance. So, bacterial epidemiology. So one of the first things you have is, how do you know whether or not you've got the same bacterium? And there has been, over the years, many different approaches to typing of bacteria. From serotyping, where you're looking at reactions between surface antigens and antisera to try to identify how similar two bacteria are. For a long time, PFGE was the pulse field gel electrophoresis was method of choice, where you were looking at, you extracted the whole genome, digested it with restriction enzymes, and then looked at the size of the fragments produced. As we moved into the molecular age and the sequencing age, there was increasing use of multi-locus sequence typing, where for each organism, they had a selection of somewhere between 6 or 10 housekeeping genes. They'd pull them out of the genome, amplify them, sequence them, look at what types of, and then based on those sequences, sort your bacteria into different types, or in some cases, just presence or absence of different genes. And then these days, we're moving increasingly into whole genome sequencing. And this is one where the field is still developing very rapidly, whereas it has become finally financially feasible to just analyze bacteria by sequencing the genome, but then you've got 1,000,001 bacterial genomes, and you need to figure out how to use that data to inform public health decisions. And as you're doing this, you have a lot of important choices. Last week the last unit was on next generation sequencing. I think you heard a lot about the different sequencing technologies, long read, short read. Even after you've done the sequencing, one of your major questions needs to be, all right, am I going to work directly with the raw reads, or are you going to assemble my genome first? Assembly in general can be helpful because it reduces file size, reduces the computational burden of comparing two organisms. But it does throw out information. It throws out information on repetitive regions. It can have problems with repeats that are slightly different. It throws out a lot of information on the quality of the sequence and how many reads are being used to generate that region of the sequence. So one of the big things you'll want to ask about any pipeline you're looking at is it working with assembled genomes, or is it working with unassembled reads? Another question is going to be, how are you actually determining the relatedness of the bacteria? I'm going to give a quick overview today of two of the most important methods, one of which is sort of alignment-based nucleotide polymorphism. That's the most intuitive of these, where you take two DNA sequences, compare them, and identify where they're different. There's also increasing use of AMR methods, which I'll be talking about in a little bit. So, as I said, SNPs, single nucleotide polymorphisms. This is a very widely used method, and it's in many ways, I think, the most intuitive, which is that you take your sequences, you line them up, you find where the DNA is the same, and you identify places where an organism might have a different base in a position within the genome. Then you look at how many of these different combinations a set of genomes have in common and use that to estimate how closely related they are to each other. And so that can be used either for establishing a phylogeny or as a whole genome multi-locus sequencing type approach, where you say, all right, I'm going to establish a set of different DNA positions where I know there are differences between different groups of bacteria, and use these to just profile quickly, this organism belongs here, this organism belongs to this group. And so in theory, this is simple. In reality, all right, someone was asking about, is there an important distinction between SNPs and SNVs? That's one where I'm not as familiar with. I think it's a terminology thing, but somebody might be able to tell me if there isn't. I think they're essentially similar, but there might be some distinctions in how they're used. But all right, so in general, this is simple. In reality, it's complicated by bacterial genome architecture, which is that bacterial genomes evolve more quickly than makes life easy. And you can have large chunks of the genome that move around. Ah, thank you. So you have bacterial genomes that can move around. You have the insertion deletions, which is a good point. Oftentimes, SNPs might not include insertion and deletion events, mainly because those tend to make the computation a little harder. Because if you have an insertion or deletion, you need to make sure you know the magnitude of it, because one insertion, if it's more than one base, can be translated by the algorithm as multiple changes, even though it was only one evolutionary event. And that's one of the things you need to keep in mind, is that if you delete a big chunk of the genome, that'll cause a lot of base pair differences. But evolutionarily, it's only one event that might happen very quickly over the course of one division. You also have regions of the genome that change more quickly than others, and other regions that are more conserved and tend to change less frequently. Or you have regions like transposons or insertion sites that change rapidly. And that's one of the big problems with bacterial epidemiology, is that bacterial genomes aren't terribly static. This is a figure that's looking at the E. coli pan genome, which is that it's asking, as you sequence more strains of E. coli, how many new genes are you discovering? And how many of those genes are exactly the same between all of your E. coli? And so you start with one bacterial genome. You've got 3,000 some genes. Next E. coli probably shares 60% of those genes, but has 30% of its genes completely different. And you keep sequencing E. coli, and you have chunks of genes that have been acquired that are different from this species, that are different from this strain to yours. And nowadays we have a large number of E. coli genes, and there's a certain number of genes that all E. coli share. That there are things that you just need to reproduce your cell, and all E. coli will have that gene. Right? You can't be an E. coli without the ability to replicate your genome and build a cell. But there's also quite a few genes in any bacterial genome that are not as essential, and one strain might have them, another strain might not. It's these areas, these islands, where genes tend to come in and out, and different strains might have different genes in there. You have plasmids that move DNA in and out. And anytime you're looking at bacterial relatedness, you have to decide how you're going to take those into account. So most SNP algorithms will have various ways. Generally, when you're looking at calculating SNPs or calculating single nucleotide variants, it really comes down to how you filter which sites are being considered and which ones are not. And also what bacterial genome you might be using as a reference for your alignment. Are you doing pairwise alignments? And then how you treat insertion and deletions and the more variable portions of the genome, and determining bacterial relatedness. So for SNPs or single nucleotide variant based methods, what's good about them? Well, they can generally use either assembled or unassembled reads, which is also true of k-mers. And they can have their resolution is really limited by the number of genomes you have. So in theory, you can compare every single base in a genome. What are some of the harder things? It tends to work better the more closely related your genomes are. As you get more differences between two genomes, it becomes more difficult to align them, more difficult to decide, is this a single base pair change or a recombination event? Your reference genome selection tends to be critical, and it's usually considering primarily the regions of the genome that can be aligned, and the variable regions of the genome are not as used to the same extent in determining relatedness. Does that make sense? All right. I clicked off that. Oops. All right, next is K-mer. K-mer is a lot less intuitive to understand. K-mers tend to be, is a set of approaches that's been seeing increasing use, mainly because it simplifies your computational problems quite a bit. So it's less intuitive, but for K-mers, what you do is you chop your genome into what they call K-mers of specified length. So for example, you have a sequence and you chop it into 4 base K-mers and you say, all right, this one has these 4 bases. Sequence 2, you do the same thing, and then you ask how many of those K-mers are shared or not shared. So in this case, you have 1 base pair difference. You can see that translates into this set of K-mers before the difference being shared between both. This set of K-mers, these set of 4 K-mers are not shared. And you can use how many of those what K-mers are shared and how many of them are shared to help evaluate the relatedness of different genomes. And so these approaches, they've got some advantages in that they don't require genome assembly or alignment to start with. It's just asking, if I chop my sequence up into an array of K-mers, how many of them are shared, how many are not. They don't require a single reference. They're much less computationally demanding searches. Now, you do lose some phylogenetic resolution compared to alignment, and it doesn't provide any information on the nature of those shared or differential regions. You do a SNP analysis and you've already aligned the genomes, and you can fairly easily immediately start asking, all right, where are these differences and what kinds of genes are these differences in? Whereas with K-mers, that's going to be a separate analysis. So how are some of the ways that this can play out? For this, I just pulled up, as a use case, I pulled up the NCBI pathogen detection pipeline, which was designed to try and quickly analyze new genome sequences as they came in to compare them to the existing database of pathogens that have been detected. So I kind of like this because they use basically every single one of these approaches. So the first step in the pathogen detection pipeline is to assemble the genomes using a K-mer-based assembly algorithm. But they found that that assembly algorithm often didn't do a good job on identifying antibiotic resistance genes. So to supplement that and improve their resolution of antibiotic resistance genes in particular, they also have a separate reference-guided enrichment and assembly step to make sure that they aren't missing too many antibiotic resistance genes. And after they've assembled the genome, they do a surface level identification. And if it's an organism that has greater than 1,000 isolates in the database, they have already pre-calculated a SNP-based sequence typing scheme that allows them to pre-cluster each new genome that falls within that cluster into sequence types. For organisms with fewer than 1,000 isolates, they'll just do a KMER-based clustering step. And then once they have these clusters, within each cluster, they choose a reference sequence within that cluster, align all genomes to that reference, calculate within cluster SNPs for their own scheme, and use that to build a phylogenetic tree of the cluster. So a little bit of talk on strengths and weaknesses. So one of the strengths of bringing in using a whole genome-based epidemiology pipeline. Well, in theory, you can do a single laboratory protocol for many different species, right? You generate a library and sequence it. And that's pretty much the same thing for every bacterial species. And in that one step, you capture a massive amount of different classification information. You can use the genome to calculate species, phylogroup, sequence type, SNPs, all of that simultaneously. But there's also a few weaknesses. One of which is that you always have to keep in mind how good your sequence quality is, how good your assembly quality is, what references are you using to do your comparisons. There's no, and then the next two are kind of the same thing. Well, we also, basically every database, every group will often have their own analytical pipeline with their own decisions, and those will all make their data not interoperable. And that's one of the other big things, is that oftentimes to do a new analysis, we don't have a consistent across the field terminology. So there's a lot of issues with cross data set comparability. You almost have to recompute your data set for every new group of sequences that you bring in. All right. So that's basically my 1,000-mile overview of bacterial epidemiology and whole genome sequencing. Next problem is how you actually find your antibiotic resistance genes. So I'm going to be moving a little more quickly through here. For a long time we identified, so when you're looking at antibiotic resistance, you tend to have multiple data, one of which is the phenotyping, often accomplished through MIC testing, which tells you which antibiotics are able to kill the bacteria or inhibit its growth, and at what concentrations. There might be PCR-based antibiotic resistance typing, where you have an assay to target specific antibiotic resistance genes, ask, does this bacterium have it or not? There's been a lot of use of probe-based methods over the years. And then again, whole genome sequencing can, in theory, tell us a lot about antibiotic resistance. So when you're how do you find an antibiotic resistance gene in a genome? So one of the things to look at when you're looking at methods is, how is it doing its search? What databases are you comparing your sequence to? How well does that database cover my organism? Is it really designed to identify antibiotic resistance genes in whatever bacterium you're working with? And how good is it to pick out rarer genes or point mutations? And this is one where, again, the field has 1,000,001 different programs and databases and methods of identifying antibiotic resistance genes. These days, the most prominent, I think, is AMR Finder Plus. Resistance gene identifier was one that came with the card database, but there's a million and one tools that have seen use many times, are different levels of popularity. It's difficult to keep track of which ones are the favorites at the moment. And then on top of that, you have not only what program are you using, what search method are you using, but also what database you are using, because all of it comes down to these curated databases where people have identified genes and associated them with antibiotic resistance. With some of the most prominent being CARD, the Comprehensive Antibiotic Resistance Gene Database. You have the reference gene catalog on NCBI. A lot of the different antibiotic resistance pipelines came with their own databases, which are often permutations of CARD plus or combinations of different databases that they might add in. And then you have, how are you searching the database? How are you comparing your sequence to that database? Here, the major tools are BLAST or other homology-based searches, where you're taking the sequence, translating it, comparing it to other sequences in the database, looking at alignment. In this case, one of your major issues is that you have to decide what cutoff you're using, what significance cutoff you're using to determine, is this a match or not? HMM-based methods, those are ones where people have pre-calculated gene models that look like this, where they're saying, all right, for this gene, most versions of it have a P here, a G here. This one's more variable. These are highly variable. This one, most of the time you see an S, sometimes you see an A, right? And these have the advantage that they can often recognize more distant sequences because they're emphasizing more heavily similarity at key points in an active site, key points in the gene. And when somebody is developing the HMM model, those are usually have built into the model a cutoff for, do I count this as a match or not? As you look into antibiotic resistance, one of the other things you need to consider is things like BLAST and HMM are really good at identifying acquired resistance in the form of entire genes that move. So things like a lot of the prominent beta-lactamase genes that move around on plasmids, it's really easy to find those. You get into other types of antibiotic resistances, point mutations, where just changing, for example, the gyre gene in E. coli can cause quinolone resistance. That's a lot harder to recognize. It requires tools that are specifically directed at identifying point mutations, because most times if you're doing a BLAST gene, a BLAST search is really great at saying this is a gyre gene and not so good at saying this is a gyre gene with this particular point mutation that makes it resistant to antibiotics. And then when you start getting into other kinds of mechanisms of resistance, such as gene amplification, where a region of the genome with a gene that produces some resistance gets duplicated so that gene is produced more, which then confers full resistance, that's very difficult to detect through database searches. Regulatory mutations, in theory, you can pick them up. In reality, they're often sort of point mutations, and you should be able to pick them up. But in reality, most searches are actually protein-based. They focus on the coding regions of genes and might ignore the regulatory regions. And so those will often be difficult to detect through regular antibiotic resistance gene databases. And so I'm not going to go into this in depth, but this is how NCBI looks for antibiotic resistance genes, where they've got a whole flow chart of methods. They do protein blasts, they have HMM searches, they ask, do they agree or not? Is it a gene that's marked as an allele where it's a point mutation? All right, now we need to run through a special program to ask, all right, which version of GIRA is it? Is it the resistant version or the not resistant version? Is the gene in a small contag and not complete? All right, now we need to do a special search to try to figure out, do we believe this match or not? And so all of these together are put together to try and give you a report on what antibiotic resistance genes are and are not present in the genome. And whatever pipeline you're using, you need to sort of think through all those different options for, is it a partial gene because there's a contag break halfway through? Am I using the right method to pick up point mutations, which often require organism-specific tools? Are there organism-specific tools for my bug to detect point mutations, or is that just impossible? Or is the database just not there? So in general, strengths, we're really good at identifying horizontally transferred, well-known antibiotic resistance genes. We're pretty good at identifying horizontal gene transfer events. We're okay at finding point mutations in well-known organisms, not so good at finding point mutations in non-model species, and really pretty bad at identifying things like regulatory mutations, gene amplification events that are more edge cases for detecting resistance. All right, and then we're running low on time, so I'm going to do a 50-mile-an-hour breeze through on mobile genetic elements because these tend to be more specialized searches. So if we want to ask, was my gene acquired by antibiotic resistance or not? One of the first questions we ask is it on a plasmid? Where plasmids are extra chromosomal DNA elements with the ability to replicate. So on their own, so they're essentially second chromosomes. They can come in pretty much any size. They're often circular, but not always. Generally, when we're classifying them, what you look at is the origin of replication. What defines you as a plasmid is the ability to replicate yourself. And that means that you need to have an origin of replication. Starting in the old days, they discovered you can't have two plasmids the same origin of replication in the same cell. So classifying plasmids became a matter of asking, can these two plasmids replicate in the same cell or not? And so that's why plasmids are often classified these days by incompatibility groups. These days, we don't do those tests anymore. We just look at the origin of replication and ask which group does it fall in. But plasmids can also have multiple replicons present in them. And then to make your life really difficult, sometimes the plasmid is standing on its own, but sometimes the plasmid can get incorporated into the genome. And I already mentioned conjugation. Plasmids are important because they're particularly good at moving between bacteria and moving between species. And then DNA moves around within cells. We did a very brief mention of transposons, insertion sequences. There's a lot of ways for genes to move between plasmids and the genome, for plasmids to incorporate into the genome, for genes to move between plasmids, build up multiple gene resistance cassettes. And a lot of these you're going to have to do some more in-depth genome analysis to try to figure out, has this happened? What is going on? And so in general, detection of mobile genetic elements requires very targeted tools. You'll have tools that are specifically designed to detect plasmid origins of replication, detect transposase genes and insertion sequences, can detect integrons, And then figuring out whether or not these things were involved in the movement of AR genes often requires gene neighborhood analysis. How close is this origin of replication to my gene? Is it on the same contag or not? If it's not on the same contag, and that becomes difficult because that depends a lot on your assembly quality, and oftentimes mobile genetic elements don't assemble particularly well, particularly out of short reads. And so it tends to be one of those guilt by association things where if you've got a plasmid origin of replication right next to your antibiotic resistance gene, which is on a circularized a contag that you can see as circular, great. You're reasonably certain that this antibiotic resistant gene is on a plasmid. If it's on a big chromosomal section, what looks like chromosomal DNA, but there's a plasmid origin of replication 10 kb away, maybe it's an integrated plasmid, maybe it's not. You're going to have to do a lot more looking. If your antibiotic resistance gene is just on a contag on its own where it hasn't assembled with the rest of the genome, you're never going to be able to tell. And so this is one of those where, again, the more you go into there, the more difficult the detection methods get. And then finally, you have to consider, as I said, things like sequence quality, depth, assembly quality, how long are your contags, how good are the contags that is the assembly part that your antibiotic resistance gene is on. And then the things that we don't tend to think about when you're doing bioinformatics are things like isolate handling. How many times was the cell passage before it was sequenced? Was it kept on selective media the whole time, or did it transfer through a non-selective media, at which point sometimes the plasmid will just get booted out of the cell and you lose the resistance gene. And so now you've got a genome sequence that doesn't have a resistance gene, even though you know the bacterium is resistant. Is it because it is a resistance gene that you don't identify, or did you just lose the plasmid during passaging and your isolate no longer matches whatever came out of your patient? There's all the other aspects like DNA extraction methods and library construction methods. It's become, I think the field has gotten more aware of it, so most of the tools are less likely to discriminate against plasmids. But some of the earlier DNA extraction methods, some of the library construction methods would discriminate against smaller circular DNA pieces like plasmids, and those would often then not even be sequenced. Or depending on how you filter your contigs, if you filter contigs based on read depth, it can be easy to throw away plasmids. Some assembly methods don't like plasmids. These can all impact whether or not your mobile genetic elements even then show up in the data you're trying to analyze. So at this point, I am running late, so I had a brief overview talking about some of the ways working with real data and how these different things can show up. But instead, I am going to cut off here so that people can get to their next things and or ask questions if they've got a few extra minutes.